In a previous video, we talked about cardiac pacemaker cells, which are one of two types of cells in your heart. Their job is to initiate action potentials throughout the rest of your heart, specifically in cardiac muscle cells. And that's what this video is going to talk about. We're going to talk about cardiac muscle cells and how they do their job of contracting so that blood is pumped throughout your body. And so here we have several outcomes that we're trying to get through. And it may look like a lot, and this is probably the video that has the most of any outcomes, but they really tie together very well, and it's not as daunting as it might look. Now to start off, we've got this figure. And on the right side, we've got an action potential for a cardiac muscle cell. On the left side, we've got an action potential for a skeletal muscle cell. Now, I want to remind you, we haven't talked about, and we won't talk about, an actual action potential for a skeletal muscle cell. But I just want you to look to see how it's different from a cardiac muscle cell. And there's two things that stand out. One, they just look completely different. So the top panels are the actual action potential. And then on the bottom, we have force production over time. And as you can see, it takes a lot longer for a cardiac muscle cell to actually undergo an action potential. And we'll talk about why this is in a minute. Okay, so here's a blown up view of that cardiac muscle cell action potential. And so as you look, there are 5 phases here that we're going to talk about. But the big thing that's important is this plateau. And we'll talk about what causes this plateau and the significance of this plateau in a couple of minutes as well. But it's this plateau that actually causes the length of an action potential in a cardiac muscle cell to be longer than a skeletal muscle cell action potential or even a cardiac pacemaker cell. So when it comes to a cardiac muscle cell, there are multiple action potential phases. We have depolarization, which is caused by sodium influx. Then we have what's called early repolarization caused by potassium efflux. And potassium is actually leaving the cell for quite a while. But in the middle of this, we have the plateau. And so that potassium efflux continues, but during the plateau, we also have calcium going into the cell. And so this essentially balances out or maintains for a short period of time the membrane potential of these cardiac muscle cells. Once those calcium gates closed, then repolarization continues due to that potassium efflux. And then we get back down to resting membrane potential. And this is also due to potassium efflux. And so let's go through this action potential and use those permeability curves that we used with the cardiac pacemaker cell. Now remember, these permeability curves are just giving you an idea of how easy it is for a specific ion to flow into or out of, in this case, a cardiac muscle cell. So during depolarization, the permeability of sodium spikes, okay? And so we can see that right here. So notice that we've got a high level of permeability for sodium, whereas potassium is actually decreasing in its permeability. So as a result, membrane potential becomes less negative quite rapidly. Then we have this early repolarization. And so what's happening actually is that there's some potassium channels that are still open from when we were undergoing depolarization. Then what happens during plateau is another set of potassium channels opens, and you're not going to ever have to differentiate between the two types of potassium channels. Additionally, we have calcium channels that open. So as a result, we have calcium going in and potassium going out. And they essentially balance each other out. If anything, there's still more potassium leaving than calcium going in, and so it's more of a downhill as opposed to a plateau, but it's a much gentler slope. And so why do we have this plateau? Okay, ultimately, the purpose of the plateau is to extend the time of that action potential. And this is key because what it does is a couple of things. One, it prevents tetanus. And what we mean by tetanus, and we talked about this during the muscular system, is a sustained maximal contraction of the muscle. Okay, just think about your heart. If it's constantly contracted, what that means is that it's unable to relax and then it can't fill up. And so this plateau gives the cardiac muscle cells time to relax so that blood can enter into the chambers. And we're generally focused on the ventricles, so this allows the ventricles to contract and relax, and while they're relaxing, blood can fill up so that we can have an efficient contraction on the next time. And so from there we go to resting membrane potential, repolarization and resting potential. And as you can see, both of these are caused by potassium channels. Okay, so potassium is leaving the cell, And basically what's happening during resting membrane potential is that potassium's leaking out slowly. It's close to what we would call its equilibrium potential. So it's not leaving rapidly, but it's basically maintaining that resting membrane potential. And you're not going to need to know a value for that resting membrane potential. So if we look at it all together, yes, it's quite messy, but you can still kind of see, look for where the spikes are. Okay, so here, depolarization is sodium. You have some potassium here, which corresponds with this early repolarization. During plateau, you have calcium and potassium, and then during repolarization, it's all potassium. So this leads us back to calcium. If you remember with the muscular system, we always joked that the answer was cross bridges, and cross bridges are formed because of calcium. The same is true when it comes to cardiac muscle cell. Okay? And so both of these, both skeletal muscle and cardiac muscle, contract based on cross-bridge cycling, or the sliding filament theory. And so we need calcium. So calcium is very important. Now, calcium in a cardiac muscle cell enters the sarcoplasmic reticulum via the ryanodine pathway that you're already familiar with, and the IP3 pathway, which you're also already familiar with if you remember back to our G protein discussion. Okay? But we have to remember that we have SERCA. Just like the skeletal muscle has a calcium ATPase pump, so does cardiac muscle cell. And so what this pump does, as a reminder, is it takes calcium that's in the sarcoplasm and brings it back into the sarcoplasmic reticulum, which allows that muscle cell to relax. So as a result, there's less calcium, less cross bridges, less force. In other words, the cardiac muscle cell is able to relax. So let's review these pathways really quickly. So the ryanodine pathway, action potential activates those voltage-gated calcium receptors that are on the DHP, or on the T-tubule. Okay? Ultimately, this activates ryanodine receptors on the sarcoplasmic reticulum. Calcium leaves the sarcoplasmic reticulum and enters the sarcoplasm. Okay? Calcium binds to troponin. Actin and myosin are able to bind, forming cross bridges, more force. This is the primary pathway to get calcium to ultimately help with cross bridges. Then we've got this G-alpha-Q pathway. Okay? And so we activate G-alpha-Q, which activates PLC. PLC converts PIP2 into DAG and IP3. And what we care about here is IP3. We're not worried about DAG. And so IP3 also opens some calcium channels on the sarcoplasmic reticulum. So calcium combines to troponin, more cross bridges, more force. This isn't as important, doesn't play as large of a role in cardiac muscle cell contraction, but it is there. So take a minute, look at these three figures. So on the top left, we have a cardiac pacemaker cell. On the top right, we have a cardiac muscle cell. And on the bottom, we have a skeletal muscle cell action potential. They are all very different. Okay, and so just look at what's similar and what's different. Kind of the main things that are similar: they both depolarize, they both repolarize, but other than that, there's a lot of things that are different. So, and we can summarize it with this table here. And so take a moment to look at this table, look at the ions responsible, how long it lasts, whether or not they've got a resting membrane potential, things like that. So at the end of this video, can you do these four things? Can you draw a cardiac muscle cell action potential? Can you identify the ions responsible for each phase? Can you tell yourself why we need a plateau phase, and why is calcium important?